Hello, and welcome to this live Q&A, which is part of the Oxford Sparks Science at Home campaign. I'm Aim Maidment, my pronouns are they, them, and I'm one of the communications team at the university's Zoology and Plant Sciences departments. And I'm joined here today by Rob Salguero-Gómez, who is an associate professor in ecology. Hi, Rob.
Hi, Aim. Thank you so much for having me today. I'm doing very well.
Good.
How are
I'm
you?
very glad to hear it. Yeah, I'm doing fine. Shall we jump right in at the deep end? And could you start by summarizing your research in 60 seconds as a starter for 10?
Sorry, could you say that again?
Could you start by summarizing your research for me in
Absolutely.
60 seconds?
So I am a population ecologist. What that means is that I am interested in the mechanisms that allow species to persist on this planet at the population level. As you know, all species are composed by one or more different populations. And understanding how and what makes populations increase, perhaps sometimes all too quickly, like invasive species, or decrease also quite rapidly, like some endangered species, like the Iberian lynx in Spain, is one of the key features of research and one of the main attractors of ecological theory and experimentation in my field at the moment. So that's one of the main areas of research that we are currently quite heavily focused on in the moment.
Nice. Very succinct to cover quite a large amount of things, really. One of the things that I'd like to ask you about quite specifically is how do you sum from senescence, the process of aging, which I believe some of your more recent work has been focusing on?
Absolutely. So I did give a really brief introduction to what we do in my lab in terms of science from a population ecology point of view. And one of the areas of research that we've been working on for quite a few years now is understanding how, where, and when species can escape from senescence. Senescence is a Latin word. To senesce means to age, but not just to age in the way that you and I think about becoming perhaps older by virtue of accruing more years and getting to blow candles on our birthday cakes. That process, the process where you just accrue years and you become older without any implications on your fitness, on your performance, that's what we researchers refer to as aging, with no implications with whether or not you're becoming more decrepit as you get older. Senescence, on the other hand, is a decrease in the fitness components as you get older from the time that you first become mature. So picture, for instance, a human, an individual that achieves maturity at age 16. What happens shortly after age 16 is that there will be probably a peak in fertility around age 18 or from 18 to 25, and then a sharp decline in the fertility potential. That's one of the two fitness components. That's one of the two things that natural selection selects for. What's the other thing? The other thing is survival. So it turns out that after we first mature, the likelihood that we survive declines. And it declines in humans, it declines in hominids, it declines in things that look like us. But the moment that you start to look away from our anthropocentric point of view, the moment that you start to look into things like corals, sponges, and I'll remind you, we still haven't stepped out of the animal kingdom. Corals and sponges are animals, as you know, let alone when we jump into other kingdoms like fungi or plants. Those patterns of declines in fertility with age and decline in survival, or the opposite, an increase in the risk of mortality with age, are not sustained. So in the last years, what we have found, my research group and also collaborators around the globe, is that senescence, that is that decline in fertility and that decline in survival, is not the rule, as we thought for a very long time. In fact, senescence seems to be an exception. And it's an exception that we are unfortunately not able to escape from, you and I, humans being, right? If you think about it from a point of view of what it means for societies, it means everything. When you go to school, how long you stay in school. When you graduate, how long you work for. How much money are you likely to get from a bank when you ask for a loan for that house of your dreams or for your car? How much money you've got left after you retire. All of that is based on the likelihood that you will become more decrepit at a specific rate, that rate of senescence. Well, it turns out that many other animals and the vast majority of many other organisms outside of the animal kingdom don't adhere to those rules. What we're trying to understand in my group is what are the mechanisms? What species are able to escape or to at least postpone senescence, and where in the world are they found?
Is absolutely fascinating, and again, so broad. To everybody watching out there, thank you for joining us, and remember that you can send in questions via the YouTube sidebar, and we will see if we can ask Rob to answer any of the questions that could come up from this. So you've mentioned briefly there, Rob, what senescence means and some of the impacts that it has as a societal concept. What does it mean for those species who don't experience senescence in the way that we do? And what impacts does that have?
The perception of these species, and allow me to give them names, first names and last names, so that we can contextualize. So we have species like hydras. These are tiny metazoans that are found in rivers. And they're given the name hydra after the mythological creature, the hydra. If you remember from the myth of Hercules, Hercules will cut off one head and then two new will emerge, right? So there's hydras that actually look like that. I would very much encourage the audience to look up on Google hydra, but not hydra myth or mythology, hydra lab, and then the
hydra, but the real ones.
Exactly. So things like hydras, things more attainable, like albatrosses actually, that can live for a very long life and still go strong in terms of reproduction. Things like many pine trees, actually, bristlecone pines among them, and many other species in the plant kingdom. These species have a completely different perception of time. Let's put a time mark on this. Bristlecone pines. Bristlecone pines are currently the record world Guinness of longevity for non-clonal species in the globe. There have been some individuals of bristlecone pines in the US that have been dated to be over 5,000 years. What this means is that some of these individuals, when they were not just germinating, but when they were juveniles, the Egyptian empire was in baby steps at that point. The Egyptian empire has come up and collapsed, as have many other empires, including the Roman, right? And they all have collapsed, but some individuals of this species still persevere. Understanding how they do so is really, I find fascinating, and one of the main lines of research that we're trying to conduct in my research group.
That's amazing. What are the biggest challenges that you find when doing this sort of aging research?
One of the main challenges that we find is I could boil it down into two, really. First, somewhat artificial division between kingdoms. For a very long time, aging research and senescence research has attracted a lot of attention on humans. When this attention has been expanded a little bit more, it's been expanded to things that look like us, things that have got 2 arms and 2 legs. So mostly mammals, and if you allow me the stretch there, birds as well. So we have acquired a vast amount of knowledge on the mechanisms of senescence in mammals and birds, but beyond that, we know very little. So that's one of the main limitations. The other limitation will be trying to understand what are the mechanisms that make some species become more decrepit as they get older, and other species become almost more vital, more powerful, more healthier, if you will, in species that live very long time. When our lifespans, our own lifespans are pretty limited, we tend to think of ourselves as long-lived. I would argue we're actually mid to short-lived. Our midlife expectancy in some countries ranges between 60 and 80 years, right? Well, that's nothing to a bristlecone pine. We're only talking 2 digits versus 5 digits in some cases, right? So trying to understand how species across the tree of life age, how their fitness components may be declining or increasing as they get older, is something that requires either the commitment of myself and people in my lab and people in the labs of the people that will graduate from my lab to follow through time, or some technological shortcuts. And that's something that we're currently investigating and we're very excited about as well.
Could you tell me a little bit more about those technological shortcuts briefly?
Absolutely. So there are some species that leave a mark as they're growing, right? You may be very well familiar with things like trees.
Heard of trees?
Not all of the trees. Good point. Not all of the trees. No, what I meant to say is you may be familiar with growth rings in trees.
Yes.
So not all of the trees leave growth increments as they're increasing year by year, but a lot of them do. And so what we can do is we can collect tree cores from some of these species and estimate how much and how often they've been growing into the previous years. That allows us to look at the past, the historical past of a creature that might have accrued upwards from 200 years. And from that, being able to estimate exactly how much they grew. And there's also some mathematical methods that allow us to estimate how much mortality has happened. And because we can also pick up investments on reproduction in the woods, in past reproductive events, we can also not only look at the growth of the individuals and also the chances of survival, but also investments that have been made into reproduction through time. Other shortcuts have to do with the advent of satellite technology and drone technology, for instance. What this allows us to do is not to have to spend too much time in the field, collect vast amounts of very large cover area with information that we can then very quickly take back to the lab and analyze the area.
Nice. That's really cool. I think I've got a question that I think is from the stream. Are current evolutionary hypotheses sufficient to explain senescence?
No, they're not. So this is actually one of the most hotly debated areas in the evolution of senescence. Notice that all of these theories are named theory on the evolution of senescence. No theory exists that has been named theory on the escape from senescence. And yet I'm telling you that most of the species that we have examined in my group, and I'm talking upwards from 2,000 species of animals and plants, do escape from senescence. There was a paper that was published by a researcher in the late 90s that aimed to do a review on the hypothesis on the evolution of senescence that existed at that time. And he was able to count upwards from 300. I will find the paper, the link to the paper, and I'll post it on YouTube later. So if that happened about 30 years ago, you can only imagine aging research being such an active field, how many more theories have evolved up until now, right? Still, the fact remains, not a single theory, with the exception of one that I'll cover in a second, aims to explain how senescence may be postponed, let alone escape altogether. Okay. I did mention that there was one that partly tackles this, right? So Jim Bobel, the previous director of the Max Planck Institute for Demographic Research, posed the line of thought that if fitness components are correlated with things other than age, then selection wouldn't be able to act on age. So if you're more likely to survive and to reproduce as a function of how tall you are getting, or as a function of how muscular you're getting, or as a function of something that is not truly correlated with age directly, then your fitness components will become decoupled from the age components. And that would, in principle, be able to open up the room or the door to escape from senescence. We have actually shown that in some creatures, the species that oscillate in size is a pretty good predictor of fitness components in plants, for instance. Those species that oscillate in size do have a propensity towards escape of senescence. So there's something there that is worth investigating a bit more.
Nice. Another question from the stream, which sort of talks about that fitness and reproduction. Going back to human senescence, which do you think is the most well-supported hypothesis for the evolution of menopause in humans, and why?
That's a really good question. As I said before, there's upwards from 300 theories on the evolution of senescence, and there's even more so on the evolution of menopause. There's obviously quite a few that tend to link intergenerational fitness impacts of grandmothers to mothers, and then to I think that's an area that really requires a lot more investigation. So I wouldn't dare to say which one is more accurate, what I will do is I will call for arms or more investigation in that area.
That's a very sensible reply. I guess it has to be interdisciplinary research as well, looking at the way it affects people differently. To jump away from humans again, are there any mammals that don't experience senescence?
There are. You see, the thing with senescence is that it's not a black or white. It's not whether you undergo senescence or not. Okay. Perhaps I can turn back to my whiteboard if you and the audience can see it and try to explain this. So imagine that you have got a cohort of individuals. Imagine that you start with an imaginary population with 100 individuals in it. And you're going to follow them through time. Okay? You're going to have here 100 individuals, or if I divide over 100 to make it a rate, I'll just have one. And what we're going to do is we're going to follow them from the point that they're born until they all die. Okay? We're going to call this in the y-axis survivalship.
Could you repeat that?
We're going to call what's on the y-axis, we're going to call survivorship. I don't know if you can see this very well. I'll try to bring this a bit closer. How's that? Is that better?
Yeah, that's better, yes.
Okay. And then on the x-axis, we will write H. Okay? Our historical understanding of how survivorship may change with different species was a rather black or white. The first thing that we're taught, or one of the first things that we're taught in population ecology, for those of us who took it back in college, is that there are 3 types of curves. Curve type 1, okay, curve type 2, and curve type 3. Curve type 1, what this implies is that a lot of the individuals will survive, but as they progress, they die off more frequently. Does that look familiar? That's you and I, that's humans, okay? There are other species like eagles, for instance, where the rate of mortality is more or less constant. By the way, this will be on a log scale for that to work out. And there are many other species that follow a sharp decline, so a lot of mortality, a lot of individuals being lost in early ages. And once they pass that initial bottleneck, then they will remain more or less immortal. In reality, there's nothing that is immortal. Things do die off eventually. So what we are taught in college is that things, creatures, species, do belong to 3 different boxes. You've got species that do senesce, type 1, because a lot of mortality happens at the end of life. Okay? You've got other species that potentially escape from senescence, type 3. Okay? Because once you pass the age at maturity, that's exactly from the age of maturity until everybody dies, is the phase, is the range of the demography of the species that we're examining as senescence researchers, right? And you've got another curve, which is type 2, which other colleagues have come to call negligible senescence. So you neither senesce nor escapes from senescence. But in reality, you could expect, and we do find, a myriad of different trajectories. And by the way, the trajectories don't need to be linear or curvilinear. They can go like this as well. And they do if I go like this. So to your question, which I conveniently just forgot. Can you please rephrase?
Are there any mammals that don't experience senescence?
Yeah. So to your question, I would say it's not whether you're escaping from senescence or not, but it's what's important here is to acknowledge that there's a wide diversity on the rates of senescence across the tree of life, even within mammals. There are some mammals that escape senescence, and there's even some cases within human populations where the rate of senescence can vary widely, right? So I'll give you an example. Babies being born in Japan last year have a mean life expectancy of over 100 years. Let that sit in for a second. 100 years.
Feels like a very long time.
It is a very long time. And that's, of course, already taking into account the fact that early on in life, in the life history of a human, there's high risk of mortality, right? That curve can go down quite rapidly. And even with that, there are some human societies that, they do still undergo senescence. They can live for a very long time. So understanding what are the mechanisms that allow for such wide variation in demographic trajectories is, I would argue, one of the most important questions in biology at the moment.
That's Yeah, makes a lot of sense. Another question for you. What is the best way to include senescence in population models where your species of interest doesn't have as much data available, such as UK native seaweeds?
So you have to be creative, right? The good thing about working in this era as an agent researcher is that we can create a lot of synergies with biostatisticians. So demography is, as I say, a data-hungry science. It's a science where historically, if you didn't have many individuals, and by many I'm talking upwards from 500 per population, you wouldn't be able to trace a lot of inferences in terms of how the survivorship and the fertility curves are changing with age. Luckily, there's been more models, individual-based models, integral projection models, etc., that are allowing for more flexibility, more statistical power, and a greater deal of inference. So this question it was about seaweeds, right?
Mm-hmm. UK native seaweeds.
Exactly. So to the question of seaweeds, there's also another logistical complication, and that is that if these seaweeds are permanently submerged underwater, then you have the fieldwork logistical complication. You have to get yourself certified in diving. There, I've got people in my team who are doing so, and you've got to undergo a lot of handicaps, not just the modeling handicap, right? You have to undergo the handicap of actually you can only spend max 27 minutes underwater, according to the latest PADI legislation. But the insights that you can take from working on things that are not humans or don't look like humans, I would argue, far overcome the limitations that are imposed just due to the nature of the species of interest.
Nice. That makes sense. Yeah. The question: somebody who's enjoying the talk so far, which is always lovely to hear. They can visualize how colonial organisms escape senescence as they have a turnover of individual clones. However, how do non-colonial organisms achieve senescence escape?
One of the things that is becoming more and more apparent from the research that we're conducting in my team is that there's not most likely a single mechanism. Okay. So, like I said before, over 300 theories on the evolution of senescence. There's only half a theory on the escape from it that I'm aware of. Please correct me if anybody's aware of more. I would love to read those. The escape from senescence or the postponement of senescence can come to you by, in the case of humans and some eusocial species, partitioning of labor, for instance. It's been shown that within insects, those that have a great deal of eusocial hierarchies get to live for longer. Mm-hmm. Talking about cases, excellent research being done in ants, for instance, and bees as well. In the case of non-clonal plants, you can still think of a non-clonal plant as a collection of pipes. Okay? Mm-hmm. Imagine that you have got a collection of straws that you're tying together with your hands. The way in which a non-clonal plant works is it will still have to absorb water and minerals from the grounds, right? That will be done through the uptake of the roots. Those resources will then be sent out to different shoots. Depending on how closely physically packed those pipes might be, and allow me to use my pens for this. Imagine that those are my pipes in a plant, okay? Mm-hmm. Can you see them okay?
Yep.
So if this pipe here is collecting water and minerals for a specific section of the soil, and then is really tightly packed at the trunk level, whatever resources has collected might be able to be transferred to other sections within the canopy, correct? Mm-hmm. Okay. Imagine now that we have got an architectural arrangement in such a way that the pipes are spaced out. What will happen there is that whatever resources this pipe, this xylem column, is collecting will not be transferable to other units. Okay, imagine now that whatever patch of soil this pipe is collecting resources from goes drought, or there's no nutrients enough to sustain the entire architecture. Well, that pipe will die out, but the entire remaining part of the individual will resist, right? So one of the ways in which some plants, and also some corals, by the way, and sponges manage to do so, to escape from senescence, is by internally partitioning those pipes. So hopefully you can see this thing here.
Yes.
Can you see that okay?
Yep.
Okay. This is, as you might have guessed, a cross-section from a tree. The tree is a Mallee eucalypt. This tree was collected in investigations that I was doing a couple of years ago in the Simpson Desert in Australia. And one of the striking features of this cross-section of the eucalypt is that it has a hole in it, right?
It looks a little bit like a bird to me.
Good point, good point. Perhaps let me use another one that does not look like a bird. What does this one look like to you?
Also looks like a goose flying. Maybe I've just been looking at a lot of birds.
Or something.
Yeah.
Yes. Nonetheless, what happens in this plant and in many other woody species is that as they grow in secondary growth, that is, as they're growing in width, not in height, but in width, they're adding more pipes, right? Their cambium is differentiating more zyme and more flowing vessels. But internally, the hardwood is rotting away to such a point that it might actually physiologically divide the different sections and allow them to behave more or less independent. This can happen in plants that you wouldn't initially, a priori, classify as clonal. And what this results in, effectively, is a quasi-clonal species. It's been shown that species that do this can manage to live for longer.
That's really interesting. That's very cool. Who knew all of the insides of a tree having so many answers? Another question from the stream. How do you define individuality for colonial organisms like corals or other organisms like plants and fungi?
With great difficulty, to be fully honest with you. That's how we define it.
The joy of definitions.
Exactly. I'm getting into a very epidemiological question, which is what is an individual? So demography is the science of the mechanisms and the consequences of those mechanisms on different populations, right? As I mentioned before, populations will go up or we will down or will be stable through time as a function of how those individuals in those populations contribute to it. What are ways in which individuals can contribute to it? You can contribute to a population by the mere virtue of surviving. That's really important, right? Another way in which you can contribute to a population is through reproduction. But you need to be able to, before we talk about contributions, and therefore being able to see whether a population goes up or down, we need to be able to identify the individual. So this is a topic that has kept plant demographers, plant population ecologists busy for quite a long time. And this debate, I would like to think, is now resolved. What we do is we approach this biological problem from two different but complementary perspectives. First, we evaluate the full colony as a genet. So what that means is that we will say an individual is whatever has the same amount, the same exact information of the genetic material. Okay, so everything that is a clone will be just one individual. So that's the genet point of view. Genets coming from the gene, the same genetic background, right? The same genetic information. The other approach is called the Ramet approach. Ramet comes from Latin, it means branch. So I'm already giving you a hint as to what that does, right? What this approach does is it doesn't matter whether or not you're sharing the same genetic information or not. What matters is that you may be behaving physiologically independent. Let's go back to that example of my beautiful pens. If my pipes eventually split off, they're all coming from the same genetically identical individual. So my green pipe and my orange pipe have the same genetic information unless they mutate, right? So imagine that they go independent. In a Ramet approach, what I would do is I would model those dynamics independently. A good approach, however, would take those into account. And there are mathematical methods to be able to incorporate Ramet dynamics within genetic dynamics as well.
Nice, thank you for that. Shall we move on to my brain has just got lost in my notes. Something completely different. How do you get into biology in the first place?
I do not remember the classical type of answer that I get when I'm reviewing essays for admission into an undergrad. The typical one starts with, ever since I've had conscious, I've always been involved in nature. I remember myself chasing butterflies and being amused by the wonders of nature. That simply wasn't me. I was raised a city boy, really, in the south of Spain, which doesn't have many big cities, but I did happen to be raised in quite a few big ones. I think that I was always academically driven. I think I was always curious. It could have been Greek, which I did love back in high school, or Latin, or English. It could have been math, which, by the way, I wasn't too good at. Funny enough, I now use math all the time. Or biology. And so what happened was Spain has a really interesting undergraduate system. In Spain, unlike what happens, for instance, in the US, prior to you going to college, you have to declare your major. You have to say, you know, I'm going to go to university to study topic X. And I wasn't too sure what I wanted to do. I even took an aptitude test and in my final year of high school. And when the person who analyzed my test got back to me, this person told me, you have no idea what you want to do. So I found that test to be tremendously pointless. Nonetheless, the year that I was due to apply for college, a new degree opened up in my local university, Universidad de Cádiz, Cádiz University in southern Spain, and that was environmental sciences. Environmental sciences, as you know, has a lot of different topics. So I studied chemistry, mathematics, physics, environmental law, quite a bit of biology, obviously. And it was through the time that I spent in that 5-year degree that I realized, actually, out of all the topics that I really enjoy, biology is what I like the most. And something really funny on that remark, I must say, happened on my first year. So I, you know, all throughout my high school, I had more or less gotten pretty okay grades, but the transition from high school to undergrad was really rough for me. The transition, I think, and many undergrads will hopefully sympathize with this. In my high school, we were just okay if we regurgitated facts in the exam. In college, that's not enough. You need to be able to document your line of thoughts. And that was definitely the big changing game for me when I went to college. I did really poorly in plant sciences in my first year.
And look at you now.
As it turns out. And my mother, bless her, got me a cactus as a consolation prize for my first midterm that I pretty much failed.
As a sort of reminder of these failings.
I think so. And I don't know if she did it on purpose or not, but she did create it a monster, because I just became obsessed with succulent plants. I became obsessed with desert ecology in general. And that just set me off in a record of really wanting to know more and more about not just what allows life to persist in really tough environments like deserts, but also what has allowed for them to become so biodiverse, so speciose, to have such many different strategies of going about making their living. So that's my little story.
And so it makes sense looking at just how interdisciplinary your research is nowadays. It's interesting to hear that's how you've always sort of approached it. What's your favourite part of your research or being at Oxford?
You want the short list or the long list?
Let's go for the short list today. We can come back for the long list.
So I would say that being exposed to people who really do mean to be there, students who are very eager to learn and to listen to, but not just to listen to argue with you, is really priceless. Having such a supportive collegial system, both in my college, Pembroke College, and also in the Department of Zoology, is fantastic. I have lived and worked at 7 universities, I think, the last time that I counted. I've lived in 3 different continents, Europe, North America, and Oceania. And the working environment that I have witnessed and enjoyed here in Oxford really is top-notch.
That's something that we hear time and time again, especially within zoology and plant sciences, which is such a lucky environment. Absolutely love it. I know you've got some video prepared if you'd like to show us some of the projects that you're currently working on. We'd
definitely love to. So let me see. I've got a few videos that I like to share with you. So let me see if I can share my screen. So okay, hopefully you can see this.
Technology is incredible.
Is it working?
Yes, not yet. It will be in a second. I'll let you know when we have the go ahead.
Fantastic. So let me tell you the spiel of what this video is going to show you. So one of the things that this research based on understanding the drivers of population dynamics become very prolific, species become endangered, what makes some species undergo senescence and other escape from senescence. This research has required a lot of data. Has the video come through yet? Fantastic. Yep. So I'm going to click on it. So this research has really required the investment on the collection of data for hundreds and thousands of animals and plant species, right? This work that I've been leading with colleagues from the US, from Denmark, from Germany, is now being made open access in two databases, really rich in terms of species and information. They're called comparative and comparative for plants and animals, respectively. We have got demographic information on animals and plants from around the globe, but in the collection of this data, I've realized, together with my colleagues, that there are some really big gaps of knowledge. We simply know very little about some really important endemic species of plants and animals around the globe. We know very little in terms of the demography of species in some ecosystems. Deserts is among them, which is really close to my heart, as I shared with you before. And we know some very little amount of information about specific corners of the tree of life. Another thing that has been wonderful since I moved to Oxford is just knowing that there are some pockets of economic support to allow you to go to places. So there's a funding scheme called the Oxford Africa Funds that has allowed for me to be able to take the imagery that you're looking at in there. So this is the Karoo Desert. The Karoo Desert is in the north of South Africa. It's about 5 to 6 hours by car from the Cape Town area. And what we're doing here is we're now tracking the population dynamics of some really ecologically important, scientifically unknown species, both the plants and animals, but I'll tell you a bit more about the plants. So as you can see there, so this image, this footage was taken through a drone that in it is currently recording with a regular RGB, red, green, blue filter, but it also has a built-in hyperspectral focus. And what that does is it allows you to pick up pockets of water. What do plants operate, or what does life operate on?
Water.
Water. So with a hyperspectral camera attached onto a drone, you are able to see things that you wouldn't be able to see otherwise. Perhaps you'll be able to see some shrubs. Perhaps you'll be able to see, hopefully, some beautiful agaves. This is agave divaricata, the little spiky tree that you can see in there. Things that you may not be able to see, however, and let me pause this and open a photo, is these jewels of nature called lithops. The word lithop is a Greek word. It stands for rock and face. So you can these three little pebble-looking creatures here. Hopefully you can see them. Mm-hmm. So the taxonomy group Lithop contains over 300 plant species, of which we know nothing demographically. We know nothing about the current conservation status. And what we're trying to do is to inspect how feasible the current population dynamics are. We know that they're very much environmentally driven. So if they get hit by a drought in a series of consecutive years, the populations will go locally extinct. And we also know that through the black market, actually, just like it happens with elephants and with rhinos, lithops have been poached. Some of these individuals can go up for upwards of 500 euro. They're very small, so you can imagine how much money you can pack in a luggage. And in fact, during my first investigation, when I went down to Cape Town, a European couple was caught with 2 full luggages of lithops that were illegally being moved to Europe for the black market. So this is one of the things that we're trying to understand is how are the population dynamics of lithops doing in their local habitats? And this is something that really requires you going to the field, becoming intimately acquainted with the plant species, using some technological shortcuts through the help of unmanned aerial vehicles, aka drones, and then building demographic models to help us project the impacts of poaching, the impacts of environmental change, and being able to project through time.
That's absolutely.
This is one of the lines of research that we're conducting. May I show you another one as well?
Absolutely. I think we've got time for another one.
Yep.
5 minutes more, so let's do another one quickly.
So I would say that most of the things that we do in my in my lab are computational. About 70%, perhaps a bit more of the time that my team members and myself spend doing science is in front of the computer, building models and trying to understand the abiotic and biotic drivers of senescence and other demographic trajectories. What you can see here, however, is a laboratory experiment that we're currently conducting in the Department of Zoology. So what you have here is worms, flatworms, from the taxonomic group Platyhelminthes, different species that are being fed different amounts of food. So one of the things that we're trying to test, I'll show you, I'll go back in a second. One of the things that we're trying to test with the different species of flatworms, here you can see a piece of calf liver, by the way, and you can see how they're happily being attracted to it and they're munching on it. What we're trying to see is whether caloric restriction has the benefits that we have thought of to apply to long-lived species. I'll go back and I'll make reference to some of the questions that I was asked before, I think by Liam Lack, on how do you define an individual in a clonal species. Well, it turns out that flatworms have the really cool ability to split themselves. I'm gonna pause the video and I'm gonna show you, I'm gonna try to point out here with the cursor, hopefully you can see that.
Well, it looks like it has a little tail.
Exactly, so you see that little indentation there, right? That's not what a flatworm looks like normally. In fact, this flatworm is undergoing an internal scission. In a few days, this flatworm, Gerardia tigrina, will have become two. It has the ability to split itself. It does so when things are getting tough, by the way. So whenever the amount of resources are low, it will split itself. And what we're trying to understand is what happens when you experiment with the amount, the quality, the timing, the duration, and the predictability of the resources. This work, which is led by one of the postdocs in my group, Jacques Dia, is requiring quite a bit of manipulations and a lot of patience and a lot of photography as well. So all of this data, we're hoping eventually will make it also into one of our demographic models with hopes of understanding under what conditions of food quality, amounts, timing, availability, do species start to postpone the onset of senescence.
That's fascinating. I'm going to end it with one final question, which is quite aspirational. What advice would you have for anybody watching this who would like to become a researcher or a scientist?
I think that acquiring some experience in the lab or in the computer or in the field is quite important, because the reality is that science can be quite taxing. It can be quite a tedious thing to do sometimes. It's priceless in the way that it satiates one's curiosity. But it is, of course, hard work. They don't call it field work without implying the work part of it, obviously. I think that getting some experience is useful. I think that the Department of Zoology can offer some really innovative opportunities for students in high school, for instance, to come and join us and do some shadowing exercises or even some summer exercises. That is once the current situation has been cleared. Of course. And I would also pitch a note for good grades is not everything. I think that, to me, as a person who is obviously always keen on attracting talents, the thing that I'm not particularly blinded by, and I think that many of my colleagues as well, is just getting AAA star or 100% on your GDP, on your GIRF. The perfect grades are, to me, not necessarily an early warning signal of success. I've seen people with medium grades doing really well. I've seen people with excellent grades actually struggling. So I think that the passion, having some experience, and realizing that grades are just one dimension of your persona.
Brilliant. I hope that's helpful for anyone watching who's hoping to follow in Rob's footsteps. Thank you so much for doing this talk. It's been really, really fascinating. We've covered quite a lot in 45 minutes, I think. I know there were a couple of extra questions on the live stream, so maybe you could, if you have a moment, pop in and give those an answer on YouTube. Wonderful. So a couple of programming notes before we wrap up. On Thursday, I'll be back with PhD student Holly Booth, and we're going to be talking about shark research for something a little bit different. Oxford Sparks are going to be doing Q&As like this, or Twitter takeovers, or new videos every Tuesday or Thursday for the foreseeable. and you can follow along on Facebook, Twitter and Instagram at Oxford Sparks. Thank you so much for joining us, and remember to look after yourselves and stay safe.
Thank you so much, Ian.
Thanks, bye.